We are just waiting for more folks to pop in from the waiting room. So we'll get started in a minute or so here.
Sorry, I don't do Zoom that much.
That's all right, you're doing great. Yeah, if you'd like to turn your cameras on, we'd love to see you, but you don't have to do that if you don't want to, or you don't have the internet speed for that. We will ask you all to keep yourselves muted during the program. You got it, Lori. Perfect. Good evening. If you're just joining us, we are waiting for just one or two. We just got folks popping in the waiting room. We'll get started in a second here. All right. Okay, well, I think we will go ahead and get started. Welcome everyone to our virtual science pub. My name is Audrey Eisenhower. I'm the education director here at Squam Lakes Natural Science Center. And these virtual science pubs are a partnership between the Squam Lakes Natural Science Center, the Squam Lakes Conservation Society, and the Squam Lakes Association. So we all come together to put on this series. I see Roger's here. Hi, Roger. Thanks for joining us. And not sure if Leanne is here from the SLA, but she might pop in later. But we're thrilled to see you all here joining us. And before I introduce our speaker tonight, two things I want to tell you. We are recording tonight's session. It will be up on the Squam Lakes Natural Science Center's YouTube channel. So if you want to watch it again or want to share it with someone who wasn't able to be here tonight, that will be the way to do that. And also we have one more science pub in our series for this spring, and that will take place on May 17th. And again at 6:30, that is also a Tuesday. And we have Graham Sherwood from the Gulf of Maine Research Institute that will be joining us to talk about climate change and the Gulf of Maine and the trends and things they're finding from their research there. So it should be really fascinating. I hope you will plan to join us for that one. And Heidi, I think I'm ready to turn it over to you. So we're really excited to have Heidi Holman from New Hampshire Fish and Game, one of their biologists. They're in the non-game division, to talk to us tonight about New Hampshire butterflies and climate change, and even ways that you can get involved. So please join me in welcoming Heidi.
Good evening. Thank you, Audrey. So I'm going to share my screen. Excellent. So I'm very excited to be here to talk about New Hampshire butterflies. I spend a lot of time thinking about butterflies, and it's always great to put a new lens on it. And tonight, again, climate change is how we're going to talk about these species. And in order to do that, it was recommended that maybe we follow the story of one particular butterfly. And so I've chosen a rare alpine specialist that is unique to our state of New Hampshire, called the WhiteMountain fritillary. Butterflies are being used and have been used for several decades as we started talking about climate change, to look at how this can impact wildlife. And the reason for that is there's just so many of them, and they have varying life history traits and different ways that they approach making their way from generation to generation. And they do that in a really short period of time. So that makes it easy for us to watch as they're impacted by changes in our environment in a short order. So it's a really unique taxa group for this topic. So as was mentioned, I just like to point out that I work for the Non-Game and Endangered Species Program in the Wildlife Division here at Fish and Game. And What we do is we work on conserving threatened and endangered species. That was the start of our program over 35 years ago. But since then, there's been opportunities for us to move the needle on restoration and protection of species. We have our wildlife action plan that identifies those that we think might be of special concern, that we want to do a little more research on. And ultimately, our goal is to keep common species common. So that's when we know we're really getting a good understanding of how to steward the resources around us to protect our wildlife. So the WhiteMountain fritillary. Let's dig deep and learn a little bit about this butterfly. We started doing some research here at Fish and Game in 2017, but there has been some work done by other partners as early as the 2000s. And the original work was Samuel Scudder in the late 1800s. He was a Harvard entomologist, but he was also a founder of the Appalachian Mountain Club. And he's the one that described this species originally. One thing I like to point out when we start talking about butterflies is that there are usually groups of butterflies that look the same. And so here's a few other fritillary butterflies you might find in New Hampshire, maybe in your front yard, especially if you have violets, because fritillary butterflies typically eat violets. That's as a caterpillar, that's their primary host plant. So you'll see there's some smaller fritillaries that would be like our meadow and our WhiteMountain fritillary in the bottom left. And then we have the larger, greater fritillaries, and there's several species of those as well. So the work done by Vermont Center of Eco Studies and the U.S. Forest Service, who owns a lot of the land in the alpine zone, was looking at how many butterflies there were and also what habitats they were associated with. And so they found that they tend to be in these snowbank communities. So these are areas where the snow blows and collects. It maybe catches on crumholts, which are those short stunted trees that you see up in the alpine zone. And that snow can get very deep. It can be the head walls of some of the gulfs, like Tuckerman's Ravine, where the snow can get even up to 50 feet deep. And as you can see from these photos, as the snow melts out and things start opening up in the peaks, these are the last places to lose that snow. Sometimes it can be June or even into July. So here's some images of habitat as well without the snow. The one in the top left would be very similar to the photos before, but also there's other wetlands up in the alpine zone. There's streams or rills, and there's also springs and seeps. And one thing that we know is these areas tend to have a higher density of flowers. So things that will attract an adult butterfly when they need some nectar in that life stage. So we're uncertain if during the surveys, when they observed the adult butterflies in these areas, was it because they were nectaring in the places that the flowers were available, or was it because their host plant was only living in these wet areas? And we still aren't quite sure. That was what we were looking to seek out in our early work. When we talk about threats to wildlife, normally we have the same cast of situations, which is development, so loss of habitat, conversion, fragmentation. So as habitat gets smaller, there's less connectivity and animals can't get to those remaining patches, possibly pollution, either a point source or non-point source. But when it comes to the WhiteMountain fritillary, it lives in the WhiteMountain National Forest. So it's really pretty lucky there. It doesn't have the threats of development so much. And so climate change and recreation are really what we're looking to address when we want to protect this species. So recreation is relatively simple, just continuing to use best practices in the alpine zone and not going off trail, using created trails. You'll see in this photo in the bottom, this is one of the AMC huts. And back in the 1800s, early 1900s, when they sited and built these locations, they put them where the springs were because, well, of course you want water when you're hiking. But unfortunately, that means that they also were placed right next to this habitat that the species uses. And so it's very critical that we work to maintain the activity within restricted zones around these places. In that photo, outside of the rocky pass is actually all host plants for the species. So when it comes to climate change, there are numerous ways that this can impact the WhiteMountain fritillary. Here's one example of a particular area above the alpine garden where I visited in late June, and there was still snow along that large ridge. I actually saw people skiing. There's someone skiing in that photo, which is hard to see. And that's another example, though, of people having to walk to get to that snow patch and maybe trampling some habitat. But the following year, all the snow was gone by June 17th. And so there's a lot of work being done tracking the snow when it leaves in a lot of these areas. So there's multiple pathways that climate change can impact an organism and make things a little bit difficult and change their trajectory. In particular, what we're looking at for this species is a range shift, loss of host plants, population decline. So that would be an overall retraction of the areas we know to be populated. Changes in their body condition, which then results in impacts to reproduction or dispersal, and then phenology, which we hear about all the time, some asynchrony between a plant and a particular wildlife species. And again, butterflies have usually one or a few host plants that they eat. So this relationship is really important. And then we have this other thing called Vultanism, which we'll talk about more, but that's really how many generations, how quickly do you go through your life cycle? This work that we're doing is eventually going to lead to a prediction of what is the likelihood of extinction. So we want to understand what the risk is to this species. And there's a lot of different parameters that we'll be collecting over time that feed into that type of model. Some we may have to just hypothesize and estimate. And then we can manipulate those parameters as they're projected to change in the face of climate. And then our work right now is in conjunction with the U.S. Fish and Wildlife Service. So they've decided that these particular species, this WhiteMountain fritillary and another, the WhiteMountain Arctic, are to be considered for federal listing. And so all of our work will feed into an up-to-date status assessment to help make that decision. So again, that's another level of protection, possibly into the future. So to do all of that, we need to know so many different things. And where we were starting was the most basic of things for a butterfly. It's absolutely astonishing to myself and many people that we weren't clear what the host plant was for this butterfly as a caterpillar. So we knew, again, that fritillaries typically eat violets, but the only violet that is up in the alpine zone is pretty restricted. And those population surveys showed them occupying the range from Eisenhower all the way to Madison. So it just didn't add up that that was the host plant or the only host plant for the species. So we looked into some literature, and there's been research done with other similar species. So the WhiteMountain fritillary actually evolved from a species that lives in the Arctic. And when the glacial period happened, they were here. As they retreated, they got left on these mountain peaks. And this happened in different places all over North America. And the different subspecies that then formed created their own host plant situation. So in Washington State and the Cascades, it's vaccinium. In Colorado, it's willow. They just evolved to use whatever plant was there. And so these are the family of plants that the genus of the species has been documented using across the globe. So why is this important? You know, beyond just knowing what their habitat is, because their food is a primary piece of their habitat, there's a lot of information from the research about butterfly trends and climate change that indicate butterflies that eat just one plant don't do as well in the face of climate change as those that eat multiple plants. That seems pretty intuitive. You know, if you have options and the area, so these snowbank communities, let's say, are starting to dry up because the snow is there not as long or not as deep. And so the vegetation community is starting to change, and your primary host plant disappears, your ability to use something else will determine how likely your range will retract or your numbers will decline. So in order to find out what the host plant was, the initial work was following those adult butterflies around, and whenever they landed somewhere, rushing over to see if they laid an egg. Because typically butterflies are going to lay an egg on or near their host plant, and they're all different. Fritillaries, unfortunately, tend to use a more scattered approach, but again, we're trying to find things out from scratch. So that early work was not very fruitful. But here in this photo, there is a picture of an egg that one of the biologists working with me found very early on. And so I'm going to zoom in a little bit more. Again, this is a very small vaccinium type plant, and there's the egg. So finding that and in enough numbers to establish a relationship of proximity to a plant that might be its host plant was well beyond the capacity of what we could do. And so we use our skills of captive rearing. If you've heard about the Karner Blue Butterfly Project, I've been working on that for about 17 years, raising butterflies and releasing them to recover the population in the Pine Barrens here in Concord. And so I've been able to learn a lot about how to establish a captive colony. So we have a small space up in the Mount Washington Observatory where we do this work. And we bring in the females and we collect their eggs on a daily basis. Through the course of this, we were able to document males versus females, which is really helpful. It kind of reduces your work in half because you're not bringing in males and hoping that they're going to lay eggs, and they don't. But again, just good documentation. We were never quite clear who was who. And after several years, we were finally able to get the eggs from the females. The caterpillars hatch, but what's very tricky is that the caterpillars have to be overwintered before they feed. And overwintering is the most challenging aspect of captive rearing for butterflies. You have to get the conditions just right so that their metabolic processes quiet down enough to survive that long winter snap. And so we were able to do that. And then we did these buffet style feeding studies. So this is an example of one leaf of plants from all those different families that we suspected or other people had suggested to us to feed. And this one little caterpillar just roams around and we wait to find sign. we were very excited once we got this going, and we were able to observe caterpillars going through their entire life stage. So if you're familiar, caterpillars often shed their skins, so they have different instars, and then they form a chrysalis, which is what you see all the way at the bottom right. So this was really surprising to us, though, because originally it was thought that this species took 2 entire summers to go through its life stage, so its whole life cycle. So that means that it overwintered twice before it became an adult. But what we found in the conditions in our lab is that they can actually complete their life cycle in 1 year. And the adults emerged at the same time as the wild. So that indicated that the conditions were correct. So we found that they eat a few host plants, and they love violet, which is great. And you would have thought that, but again, it's not present everywhere. It's very limited, it's very rare. But we did find that they eat some willows and some vacciniums, so bilberry, blueberry type family, but they really prefer the ones that are associated with wetlands. We did see them eating the alpine bilberry, which is pretty much ubiquitous in the alpine zone. It tends to be drier and very similar to low bush blueberry. It's not their favorite. They nibble, and caterpillars will do that in captivity if it's there, but it's not necessarily their preferred food. So multiple species, not quite sure how much preference or use of any just yet. So backing up for a second to make sure we're all on the same page. Butterfly life cycle is the same for any species: an egg, caterpillars, chrysalis, pupa, cocoon, whatever you're familiar with, and the adult butterfly. So every species does this. And what varies is how many times they do this in a year. So that's that Vultanism. And then what life stage they choose to overwinter in. And with one particular unique case here on the right, we see monarch butterflies, which actually migrate. It's very uncommon that a butterfly species habitually migrates year after year. There's a few examples of some that might shift relative to resource use. Sort of similar to some of the bird species that come from the north in the winter. So again, one of the key characteristics that really determines differences between butterfly species is when they go into diapause, which we can think of like hibernation, a time when there's very low metabolic activity. The difference is, though, diapause is a complete cessation in growth. Like it's much more halting than hibernation, and it's got a start and an end. And the diapause is triggered by some sort of environmental stimulus. Typically, we're thinking a stimulus that's associated with the end of summer, decreasing light, decreasing temperature, food starting to senesce. But it can also be during the summer in a time when it's about to get really dry and the conditions aren't good for that particular species. So again, they can do this at any life stage, including even an adult life stage. And one very important piece also is how they do this. Do they do it every single time in the exact same stage of their life? Is it always that first freshly hatched caterpillar? Is it always the egg? Is it always the adult? We call that obligatory. And that means generation after generation, the same life stage is when they enter that diapause. Or it can be facultative. And so that's when maybe they speed up and they make it to a certain point. And so the stimulus says, go ahead and complete your life cycle and try another generation. So that's a more optional. And the way I can describe that is some of the species we have here in New Hampshire, like a grassland skipper, may only have one generation per year. But that same species, if it lives in South Carolina, may have 2 or 3 generations because it doesn't get that stimulus trigger at the same time as it would here. So that's a very important piece. The Karner blue butterfly we found through different climate research is actually facultative, and usually it has 2 generations, but sometimes instead of the egg overwintering, it tries another generation, and that can be somewhat detrimental. So these caterpillars, that teeny tiny freshly hatched caterpillar overwinters in the worst recorded weather in the world. It's crazy to think that they can survive. And so these are some of the adaptations that they have that make that possible. You know, their behavior in the fact that they tuck underneath vegetation, they have dark colors that allow them to absorb solar gain and get their body temperatures up and maximize growth in the short summer. The ability to develop over multiple years. So they can choose to take 2 years to get the right amount of nutrition. And then the ability to withstand freezing. And there's multiple ways species can do it. They actually can tolerate freezing and unthawing, or you know, they may be able to reduce the ability of their bodies from freezing by creating an antifreeze. So really fascinating. So back to the how climate change pathways affect these types of characteristics. So when phenology is changing, there's asynchrony, or there's, again, multiple generations could start occurring. There could be a shift. And we know that that's a common trait that occurs for those optional diapausing species. And what this particular chart shows is it's not straightforward, though. So we talked about specialists or generalists and how one might do better in the face of climate change because they have adaptability. But now they're saying, well, it depends on what stage you overwinter on and then what type of plants you're eating. So in this graph, they're saying eggs that are species that eat woody vegetation that overwinter actually start to emerge earlier in spring than if another species overwintered as a larva or one as a chrysalis. So there's like multiple variables that are starting to determine how great climate change affects the timing of them, either early in the spring or even late in the fall. So again, can be very complex, but the point is there can be so much variation and based on multiple traits. So what we're doing to account and watch for this. So we said phenology can result in a loss of habitat if you're asynchronous with your plant. That means you might miss your food, and so you're going to die in place, and that's going to reduce your population. So you'd expect a range retraction. And so we repeated these point count surveys that occurred from Eisenhower to Madison and all along the Alpine Garden and out to Bootspur. And they were a 10-year interval from when they were last completed. And we found that there was no evidence of range retraction. So that's a good sign. That's an indication that the population is still occupying all of their habitat niches. So there's likely not been a change in the vegetation that prevents them from living there. We'll also run an estimate to get numbers of those individuals. And as we're collecting this data, we're also getting dates. So the first time an individual is seen and the last time an individual is seen. So we'll start to be able to count the days that it may be shifting in either direction. Another way that we could track this is we've created this citizen science project that if you happen to be hiking up in the presidential range in the alpine zone and you see an orange butterfly, you can snap a photo and place it in your social media and just hashtag Fritillary Finders. You can also find the Fritillary Finders project on iNaturalist, and by having people up there as our watchdogs, we may not repeat those population surveys for another 10 years, but if you report seeing a White Mountain fritillary on July 13th, halfway in between, that may trigger us to respond sooner and go check out what's happening. So the next pathway is a change in the body size of the butterfly. So some species, there may be a shift in color if they're really dark and they need to be cooler, etc. The fritillary is a pretty benign colored butterfly in its adult stage, so that doesn't seem to be something we need to watch. But there was research on the parent species that the subspecies evolved from in Greenland that showed from 1996 to 2013, the body and the size of the butterfly decreased over time. So they've already seen that occurring in the northern extremes of many other types of Arctic butterfly species. So that says that that can definitely happen here for us. And the issue with that is when your body size goes down, the females are going to produce less eggs. So that's going to reduce the population growth and the replacement of the population year over year. The other thing is that as the body size decreases, the butterfly may have less flight ability. So it may not be able to travel as far. And as it is, they're in some pretty windy weather and they have several miles along that ridge that they're living. And so that could impact the population. When we describe a population of butterflies, they're typically a metapopulation. That means they're groups of small patches of habitats with little populations that are connected. And that can vary in how connected they are, and it's a lot of magical math, but ultimately we know that there are these individual patches that have butterflies, and sometimes they may wink out and get recolonized from the nearby area. There's infrequent movement between them, which accounts for genetics and maintaining different variability, allowing them to evolve better and adapt over time. So it's a very critical component that these patches are connected. And again, this relates to if a patch is far away and it blinks out, there's no butterflies to recolonize it, that's what's going to start a range retraction of the species. So other work that we're doing to monitor this and to determine the likelihood is we're doing marker capture surveys. And this is when we write individual numbers on butterflies, and we choose an area and then we release them, and we go back every day that there's good weather for 3 to 4 weeks, and we try to recapture as many as we can. This is usually pretty effective in places that are grassy meadows or easier. It's a bit challenging to do this in the high elevation, but we did have some success in our first year last year. And what this map is showing you is all of the known locations of butterflies that were collected prior to 2019. And when we buffer those locations by 100 meters, we can see the connectivity. It'd be where all those pink dots are touching. And there's some gaps along the range of the species. But if we buffer at 300 meters, let's say we think that they can move 300 meters pretty regularly, or at least a portion of them, we see that then the majority of the population is connected. So that's what we're trying to figure out, is how connected it is to determine how healthy the metapopulation is. And our initial work showed 261 meters is the average distance. So that's pretty good. In addition to the marker capture surveys, we're backing up this assessment with DNA data. So we also collected tissue from species, different individuals across the range. So we had 10 or 12 different locations, and we collected 10 or 20 individuals at those spots. And the DNA will also show us how often that they interact with each other, that there's interbreeding, and the connectivity. So the two data sources work together. In addition, while we have butterflies in hand from the breeding, but also some of this work marking and collecting DNA, we're taking some measurements of individuals. So this will give us the baseline information on that butterfly size. These different columns stand for forewing length, forewing width. Some of them are the body size, length, and width. So the abdomen, the thorax. And we still have more work to do. We're going to continue doing a second year of DNA analysis this year. We're going to try to do some more in the field data collection to find out how many eggs individuals lay. We're going to continue feeding to understand their food resources and that trigger for diapause. You know, as we mentioned, we originally thought it took 2 years for their population, for their individuals to go from egg to adult, but we know they can do it in 1. And now we're trying to figure out what percentage does 1 year and what percentage does 2 years. The other way that we'll get at that is the DNA analysis. So we're collecting 100 samples from 2021 and 100 from 2022. We'll also be able to see the relatedness between the 2 years, and that'll give us an idea of what percentage makes it in 1 year or 2. So it's very fascinating, and that gives us some more understanding of their adaptability. So that was the story of just one butterfly species that we've had the opportunity to really start to dig in and look at those pathways that are known to affect butterflies, try to understand and preempt their disappearance from our landscape. As I mentioned, there's another endemic Arctic butterfly that lives up on the top of Mount Washington. This species eats bigelow sedge, which is that grass that's in the bottom left, and it's fairly common in open meadows like cow pasture and the Monticello lawn near Jefferson. It's fairly limited in its range because of that host plant. There was a lot of work that was done, and it is shown that they are 2 years for their life cycle, and the DNA work that was done by a PhD student confirms that the even and odd years are basically separate populations of butterflies. And so at that point, you could actually lose the even years of butterflies and only have the odd years in the future if something were to go wrong. And another example, the Karner blue butterfly, we've been working diligently to recover the population here in Concord, and we've overall seen an upward trend. I mean, insects are usually variable from year to year, so the fluctuation in this graph doesn't surprise us that much. But we do see some extreme dips in the population that occurred in association with droughts. So 2012, 11, 12, we had a pretty extreme drought. And then also in the spring of 2020 in June. And the issue there was the lupin, that its primary host plant, pretty much dried up before the last generation of caterpillars were able to complete their life cycle. We assumed that there was some direct mortality in the chrysalis stage. And then also, because the lupin was such poor quality, the number of eggs that last generation of females laid for overwintering was probably low. It's not incredibly surprising. We know that small populations are very sensitive to stochastic events, so extreme events like drought, but we know some species can withstand it, and it really has to do with all of those unique traits about them. This is some climate prediction models from the recovery plan for the species, and it shows that in the future, we only expect to see a further reduction in soil moisture, which would create that condition in 2020. And then also a change in snow depth. And what that could impact is, as the species is overwintering as an egg, the snow keeps it in a consistent temperature and relative humidity. And if there's no snow, then the egg can actually desiccate or dry out over the winter and reduce the survival. So what we can do is just continue to try to increase the population so it's larger, and when those dips occur, the species persists. But there are 100 butterfly species in New Hampshire, and there may be more in the future. We know if range shifts, some may move northward, some may retract into Canada. It's hard to say. We may start to detect more generations of species that previously only had one adult generation per year, having 2 or 3. And there's so much that we don't know. And again, we want to try to get ahead of the curve, because when something gets to the point like the Karner blue, where it actually disappeared from the state, it's so much more difficult to try to reestablish it. And so what I'm asking from people across the state is to help join in our new butterfly monitoring network. And what this is a way for us to bring all of the data together, be it an observation that you saw when you were walking in the park. You can report that to eButterfly or iNaturalist, or even in participating in standardized monitoring efforts that we're organizing across the state. So there's so many ways you can participate depending on your ability and your time commitment. And one of the ways I think that's most simplest to start is, even if you don't know much about butterflies, if you're familiar with iNaturalist, if you see a butterfly and you snap a photo on your phone or even on your camera, you can upload it to the website and it'll give you suggestions of what that species is. So you don't even have to know. And then other people will chime in and confirm the species. And at that point, it will make it available for our project that we've created in the iNaturalist platform. And so the butterfly monitoring network on iNaturalist will sweep up all the data that is a New Hampshire specific observation that has a photo and that meets a certain level of confirmation of species. And so I'm hoping to see this number increase over time. So the vision, we're doing trainings using modules and creating butterfly groups in different areas of the state where people meet once a month and they're learning the different species ahead of time. We want to train 200 people across the state in the next few years through these different organized workshops. And then we want to create 6 standardized monitoring count circles over time. So there was one established in the Baker Pond area up near Rumney in Grafton County that's been doing this for 19 years. Recently this year, we've started the Super Sanctuary Butterfly Club with the Harris Center, and we'll be running the first point count in July. And then also the Lake Sunapee Butterfly Club, and we'll be doing that count in July as well. And that's in conjunction with Osborne Sargent Land Trust. So we're looking to join in the next year with Audubon for a capital area butterfly club, the NOAA Estuarine Research Reserve, which is Fish and Game's partners, the Sandy Point Education Center. We'll have the Great Bay Butterfly Club. And I'm certainly hoping that perhaps I can attract some interest here in the Lakes region, perhaps with the help of Squam Lakes, or possibly Tin Mountain Conservancy for the Mount Washington Valley. So stay tuned for those upcoming announcements. And we usually put those through Nature Groupie, if you're familiar, through Cooperative Extension. In addition, I'm hoping to see an increase in just the anecdotal observations that people submit to iNaturalist or eButterfly. And then we will be looking to fund a website that has all of the training modules, platforms to show the results of these different circles, and just connect us all about how we're doing monitoring our 100+ species in the state. So with that, I just want to point back to partners that are working on the WhiteMountain Fritillary project in particular. Whenever we start such an in-depth project, we need partners to really support us for the lab space, the auto road for access. We have several universities helping with postdocs and masters at this point, and the Fish and Wildlife Service, of course. And that's what I have for tonight.
Thanks so much, Heidi. Do you, perfect, yeah, bring us back. So thank you, that was fascinating. I can't still imagine how those butterflies survive up there on top of Mount Washington. But we would love to take some questions from folks. So we can do this in two different ways. One is you can unmute yourself and ask your question and Heidi will respond, or you can type your question in the chat and I'll pass it along to Heidi. So we will start that way because we have a question that came in from Eileen, and she's wondering, how do you handle the butterflies without harming them?
So typically I train people for about a week ahead of time. We work with more general butterflies to make sure that we're not impacting our rare species. We net them, and then basically we use forceps, so that minimizes our skin contact, which can rub off some of the scales. But each butterfly species varies in how delicate those scales are. Monarchs are very tough. You figure they fly to Mexico. So we practice, and we then use the forceps and the Sharpie markers, and then let them go.
Delicate procedure there. Dennis is wondering if we have an estimate, or if you have an estimate of the WhiteMountain fritillary population size.
So the estimate that came from the Vermont Center of Eco Study work in 12 and 13 was 1,500 butterflies. And again, until we complete the analysis, we can't say for sure, but there was no range retraction. So I'm not expecting a significant decrease from the work that we did. One thing you'll note that the recovery goal of 3,000 for Karner blues isn't enough to protect that species, but for somehow, the WhiteMountain fritillary can persist with 1,500, possibly into perpetuity.
Linda, you might need to give me a little extra clarification on this, but she's asked how many overwinter in New Hampshire. And I don't know if you mean species, how many butterfly species overwinter in New Hampshire?
If that's the question, I can guess. I would say probably 90%. I don't know for sure, mostly because things like the American lady, the painted lady, the red admiral, there's a little bit of uncertainty about how much they move, but they think that they do move a little bit further south instead of staying in New Hampshire. A lot of times you'll find we just don't have the information because it hasn't been prioritized yet.
And that was what she was asking. So great, thank you. Missy is wondering, how do you collect DNA from a butterfly?
So ways that it's been done, you can clip a small piece of their wing, which would be similar if a bird sort of bit at it. They do get tattered in general over time, even from a rainstorm. The other thing you can do is remove a leg. So they have 6 legs, and that has a lot of DNA in it as well. So those are the main methods.
Jane, why don't you go ahead and unmute yourself?
Okay, yeah, I'm just wondering, how does anything as small as, you know, a first or second, but even a 5th instar larva survive the winter? I mean, what sorts of chemicals could be found inside that larval body that would protect them against freezing? And I think you mentioned that some of them actually freeze and rethaw. I mean, that's sort of mind-blowing. But I guess if there's not much there except gush and imaginal discs, I guess it's not like freezing and thawing an animal. I don't know.
So they do tend to change their water, like water composition, to more of an alcohol that can reduce that freezing. And really, yeah, you're talking about cells bursting, right? When water expands when it freezes. So they shift their chemistry to make it like an antifreeze. And then there's, again, that ability for some of them to freeze, but not burst their cells as well. So the mechanisms, sometimes I haven't read up on it as of late. And sometimes I forget exactly how that goes, but yes, so there's some that can super cool up to like -50 and some that can only go to like -10, but
Wow,
pretty impressive.
amazing. Thank you.
Yeah, and if anyone else wants to jump in and ask a question, feel free to use the raise hand like Jane did, or you can just unmute yourself and pop out. I can't see you all at once here, but I did have another interesting question from Dennis. He wants to know if you're continuing the captive breeding to establish a population that you can release to perhaps fill in the gaps in the connectivity of the populations.
So definitely we're getting the protocols down so we know we can captive rear if we determine there is a rescue effort needed. And often that's not the case. Many times you're under the gun and you're doing it simultaneously. So that's good. Hopefully we'll have it down. There is the possibility, again, now that we know it can complete its life cycle in 1 year or 2, that we could somehow rescue an even or odd year through captive rearing in a way that we might not have anticipated. Yes, definitely recolonizing if there's a range retraction. We have lots of opportunity to us as long as we have the tool in the toolbox. No plans yet, though.
Mary is wondering, what is the pollard related effort for the future?
So I'm trying to take a measured approach to the engagement of what we can accomplish as citizens until we determine how excited everyone is. And so the pollard transect approach is been done in some states where we actually, you can coordinate volunteers to choose a particular park or area where they want to do a meandering transect 5 times a year. And what that does is it allows you to document all butterfly species from spring to fall, but you have to do it consistently year after year and in multiple places across the state. So it's a pretty sophisticated volunteer effort. And I would love that we got so enthusiastic about butterflies here in New Hampshire that maybe we could achieve that in the future.
And I'll just put in, a plug. Heidi mentioned that they're working to build some of this up. So if there's folks out there that are interested in getting involved, I'm going to volunteer Carol to be the recipient of some contact information. I don't know if Carol, you can maybe put your email in the chat. That would be great. But we love to see what the interest is if we can, get some folks trained and do some, participate in with New Hampshire Fish and Game and some of this monitoring here at the Science Center, or maybe partner with the Squam Lakes Conservation Society and think about some of the properties that they have under conservation as well. So if this is piquing your interest and you want to get more involved, Carol just put her email in the chat. So let her know and she'll sort of collect, and we'll see what we can do. We might start small with some education and some iNaturalist work or something like that, but it would be great to build that up. Okay, Laura has a question. She is wondering if you could just talk a little bit more about the range of the WhiteMountain fritillary. She's wondering how far south does that reach?
So it only lives in the alpine zone of the presidential range. It's very specific. It lives over 4,500 feet in elevation. And that's the only place that mirrors where the species originated from, again, the high Arctic tundra. So I get asked a lot, like, could it possibly have existed on Lincoln and Lafayette? I think there's a small patch of alpine zone over there, but it's so isolated that I have a hard time believing it's there at this point, but I always welcome sightings from people. So again, it's very restricted to that 17 miles along the presidential range from Eisenhower to Madison.
Right. Anyone else have any questions for Heidi? Great. So Heidi, thank you so much for joining us. That was really fascinating. I learned a lot about what those little caterpillars go through. Oh, there's a question. A couple more. All right. I just need to give you some more time. Mary has a question for us. Will an increase in human population in New Hampshire and associated environmental impacts, more cars, pollution, wood burning, impact our butterfly populations?
I am assuming it will. I mean, those things contribute to loss of habitat. And so if we have other species that live in restricted areas, we would want to be careful we weren't developing their last habitat. A lot of the factors you talked about could change environmental characteristics, feed into climate change. And as we now know in depth, that can manifest in a variety of ways. Sometimes it can be positive. There's going to be climate winners. And I think we always hear that, those ones that have more generations and expand their ranges. So it just, a lot of times we're saying it's going to be a shift in these community types.
Okay.
Dennis, did you have a question?
Did you mean me?
Go ahead, yes.
Okay. What my question was, when you pointed out the egg from the butterfly, do they lay multiple eggs or just one?
From what we see, they were laying one. So we've only observed it twice in the wild, and she laid one egg. In captivity, they're doing some strange things, and they will dump a cluster of eggs. And I think it just has to do with us not getting everything perfectly right. I think fritillaries in general, from my knowledge, disperse their eggs one at a time over a broad area. And what was so fascinating to me was some of those caterpillars lived for like 10 days the next spring and summer before it even started eating. So that says to me that that caterpillar has plenty of time to get to its host plant, even if it wasn't laid right next to it, which is very interesting.
Thank you.
Mhm.
Yeah, I'm just pausing to see if we have any other. All right, Jane. Oh, you're still muted.
If the ones in captivity will deposit multiple eggs at one time, do all of them wind up being viable, fertilized and viable?
What we find is the female is either fertilized or not when we bring her in. And it's hard for us to tell. We do try to, look for ones that are a little bit more worn with a swollen abdomen. We don't have much luck mating them in captivity. We have tried that. So usually I think it was 70 to 80% of eggs from a female hatched. So the majority of them are fertilized. Maybe there's just some that weren't completely formed, at some point. And they were laying upwards, some of them 60 or 80 eggs, which is pretty good. I mean, Karner blues can lay 200 in captivity. And
Wow.
I've heard of other species that can lay 1,000. There's still some things we don't know. Females may be active a little bit later than males. They may actually prefer to delay egg laying until late August to shorten the time that larva is out. But maybe because of the captive environment, they're just dumping them after like 9 or 10 days. So we're still trying to work that out. We can start bringing them in later. We can manipulate the lighting, and maybe we'll get a little bit better understanding about what's going on.
All right. Well, thank you again, Heidi, for joining. Oh, Missy.
Just one more question. Are birds the only predators?
No. So a lot of times there's wasps that will lay their eggs in a caterpillar's body, and then it'll sort of take over like Alien. It's pretty gross. Spiders will also trap adults in their webs or eat other life stages. Other caterpillars can eat caterpillars. So one thing I'm curious about is, as climate change is occurring, there could be blueberry pest species that eat that vaccinium that could suddenly move into these patches, and then you've got competition, or maybe there's a pathogen that they carry. That we could think of a million things that could go wrong, but that if caterpillars are restricted in the amount of food, we've seen this in captivity, they'll actually eat another caterpillar or a chrysalis.
Okay, thank you.
Mhm.
Dangerous world out there.
Okay, I think we're, I appreciate all the questions. Those were some great questions, and it was wonderful to have you all with us tonight. Thank you so much. Join us again in May. We look forward to seeing you then. Thank you, Heidi. Have a great night.
Thank you.